Okay, so today we will be discussing about process development, right? So any drug that we develop, the process is very important, and that's what we are going to do. So what kind of drug that we have seen now? Starting from small molecule. Small molecule means like a organic or medicinal chemistry derived molecules of few hundred Daltons that one can synthesize using organic chemistry tools and used as a oral dosage, like it's a tablet, capsule and all those small molecules. We looked at the next is a biologics drug, which can include peptides, recombinant proteins, monoclonal antibody, bispecific monoclonal antibody or antibody drug conjugate. So what are bispecific antibody? Two different epitope binding site and it can be targeting two different targets. What is antibody drug conjugate? So it's we have an antibody and chemically link a drug. And these are nowadays used for precision therapy, right? Surgical strike करने के लिए these are used. So you first it recognizes the target, the second one is a warhead that kills at this target. So antibody drug conjugate, even in our Jane Vision, like there was a talk on antibody drug conjugate, right? So antibody drug conjugates are used for precision therapy. Drug is a like it's a chemotherapy drug. So first identify where to bind, which targets, which are the epitopes on a cancerous cell. The antibody will recognize that and then drug will go and kill that cell. That's the concept of antibody drug conjugate or precision therapy. Also need to develop a platform for manufacturing these different kind of drugs. Next, drugs like a gene therapy we already discussed. The viral vectors is required, like non-viral vectors, siRNA, short RNA, miRNA, all these are mRNA, these are required. There's cell therapy or gene therapy or gene editing. So these are biologics drugs that are there, and each of the drug manufacturing requires a different kind of platform. Where these drugs are prepared and what are the protocols? So then generally they are prepared in something called GMP, good manufacturing practices. So what is good manufacturing practices? We in lab never follow any of the good manufacturing or good even laboratory practices, right? So good manufacturing practices contains of 5 important P's, right? People, right? People, that's a first. Human is the first thing in GMP. The people who are doing it, right? So comprehend role and responsibility of each of the people should be clearly defined, who is doing what and responsible for what. So responsibility of each of the person that is involved in different aspects is required and that has to be defined clearly. People is the first and most, like a first and critical requirement parameter for GMP, good manufacturing process. Why GMP is required? That ensures that product is consistent. There is no batch to batch, because we are manufacturing at large scale, manufacturing for whatever treatment of the people. You cannot say कि ये batch वाला में थोड़ा सा खराब रह गया, दूसरे batch में थोड़ा सा अच्छा. That's good for like when you do protein purification in the lab. You never too much critical about whether we are preparing each time same, 3 times, 4 times. Every time that you prepare has to be same. That's consistency needs to be developed. So that consistency has to be there, because finally we are leading to product, and product cannot have any variation. So batch to batch consistency should be there. It should be controlled and quality standard has to be made for batch to batch production. So first is a person who is doing it. Therefore, how you make sure that people are there and with different responsibility and they are producing product which is consistent, right? It's a practical problem, right? All of the people know, and we face a lot, but company also faces a lot. So let's think about a situation like Subrata is doing research in my lab and उसका MSc खत्म हो गया, he had developed some protocol, अभी वो document submit करके गया and now next person comes. Can I make sure that whatever he has done, the next guy exactly does the same? How will help me in designing a process where the next guy is doing the same thing that he has done it? Right. So the protocol that was adopted by process and protocol that was adopted by Subrata is to have the all the fine details documented properly. So next guy comes and does same. Like he has to write in the lab notebook, whether it is physical or electronic, add 10 microliter of this enzyme or this chemical. It cannot be write 10 to 20 microliter, right? Then there is a variation. So if the product is being developed, R&D में चलता है, you can vary, like do little bit of variation to optimize the process. So once it is optimized, to stick to fine defined protocol for manufacturing. So the second thing is clear specification at every phase of production. That is the second P in the GMP. Product clearly specify each of the details that we have, right? So person has, their role has to be defined, who is doing what, who is checking what, how you are making sure that things are moving forward. So person is important, then product, which is defined by or driven by the specification. Third is process, right? Process. So properly documented, like it should be simple, so that the next person can come and understand. Should not be written in a cryptic language, right? So it should be clearly written in some language which everyone can understand the follow-up. Should be written in a simple language, very clear and consistent. That's the third P, process of this. A procedure. What are the important procedure for following for the manufacturing? That has to be clearly defined. And finally, and very important, where this is getting manufactured. Premises has to be in tightly controlled. Cleanliness should be there. Equipment should be calibrated. मान लो just for an example देते हैं, pipette लिखा है 10 माइक्रोलीटर डालना है, right? You added 10 microliter, but pipette is not calibrated. Pipette 1 साल 2 साल पहले calibrate किया है. What will happen? Instead of 10 microliter, it may be 9, 8, 12, whatever. So you are adding variation. So instrument has to be periodically calibrated and authenticated that whatever it is supposed to do is working as per the specification. Then only you know this is happening. Each instrument, like whatever you are using, has to be calibrated and documented that this has been calibrated. So instrument should be there. Cleanliness has to be maintained because you are finally manufacturing a drug. Cleanliness should be there. It should be access control, right? It should not happen that whatever one guy is doing and some other guy thoda whatever comes and spoil everything. So it has to be access control. If one of you have a access for developing this thing, you should be only allowed or authorized to do that. So access control should be there and it should be very tightly regulated that the premises is there. So cleanliness and equilibrium should be there. So these are the 5 essential ingredients for any good manufacturing organization, GMP. Right? So how one can achieve? This is term called QBD, quality by design. You ensure by designing your process that the quality is ensured. So as you know, there are 5-6 phases of any drug development. First is you optimize the lead drug molecule. Then once your lead drug molecule is optimized, what is the lead molecule, what is the active pharmaceutical ingredient, you go and do preclinical trial, preclinical study. What are the preclinical study, Garima? Toxicology, PKPD, in animal model and all those data you compile. So that's a preclinical. After that, we go to phase 1, phase 2, phase 3. Once we are sure about this, we go for filing. After that, launch of the product, and after launch, you also assess post launch, what is the idea of this. So you define by this TPP, total product profile. You first define it. Then you are looking for critical quality attributes. Each of these terms we are going to define it. Then you, like these are the, say this is the critical parameter that product must have. Aggregation state, higher order structural state, binding, immunogenicity, all these are critical quality attribute. Total product profile is like what all the way from product from design to the development, what is the total product profile. Then product design space, how we are going to design it, process design space, what are the control strategy that we are going to adopt, and the process validation, like you validate it, what process we are following, and finally comes to the filing. And then you monitor it routinely that whatever we are manufacturing is following the same thing or not. So you can do this by your risk management, risk assessment, and what are the risks, kaha samasya aa sakti hai, that's risk management we are going to do. And most important thing, you have to ensure that your raw material that you are using is of good quality and consistent quality. There should not be even any variation in the raw material that you use. But we are making finally a finished product. Finished product quality depends upon what is the raw material you are using, alright. So you know, I will tell you some practical life problem that we face in academics. Suppose I have optimized, you as a student optimize for developing something, say protein production. I am just giving you not a product, even protein production, right. Now your finance officer comes and tells it, okay, why you want to buy this chemical from Sigma? JEM pe kharid lo na? JEM you know? Nahi, you don't know. Your supervisor never asked to venture into JEM. JEM is government e-tendering market, whatever. So there are all they are selling equipment, some consumables and all those. Anyone can sell it if the product is sold. Now Sigma of course will be expensive because they are whatever manufactured with a tight control and lots of profit, and somebody is manufacturing say somewhere, and you are not confident about this product. So whatever raw material that you are purchasing, you are not confident about that. So what will happen to your production of protein? It will be affected because you are not sure about the quality. First you have to make sure the quality remains same. And when you are manufacturing a drug, you cannot rely on the variations that work. But that many people don't like appreciate the pain, especially those who are in then looking the L1 price. L1 you know, right? Right? L1, what is L1? Lowest price. So if you want to buy something in government setup, you have to get 3 quotations, whosoever is L1, you have to go for that. So L1 के चक्कर में पूरा product ही बर्बाद हो सकता है. So you have to work around it. These are all government procedure. कुछ कर नहीं सकते उसमें ज़्यादा. But that's a raw material management. You have, these are real life complications. Whether you are working in an academic lab or in industry, you are going to deal with this. So start thinking about this when you really face it. So raw material management is very important. The quality, where we are keeping, what is the humidity, what is temperature and all those, because once you purchase and kept it for 1 year in some refrigerator or some corner of the lab, you are not sure whether the raw material that you have purchased, even from Sigma, purchased and kept it somewhere, you are not sure. So raw material management is very important. And then how much testing has to be done, what kind of data analysis has to be done, that also has to be thought. So DOE, design of experiment, is extremely important. Statistically, how many data points we should collect, which gives a confident result, that's important. And finally, it calls process PAT, process analytical technology, that many of the chemical engineers develop. PAT is basically how you get an optimum process developed done, and that's the thing has to be done. So what are the risk assessment and defining a specification of a limit? So you have to understand how much variations can be introduced by different process, right? Once you are manufacturing, what is the limit that we are going to set? Let's say set point limit. What is the operating range? In this range the variation can be tolerated. What is the accepted range, and what is the characterization range? This has to be very well defined for any product or process characterization study, right? So this comes from design space. What is the minimum variations or maximum variation that can be afforded? 1%, 2%, 5%, 0.1%. So because finally there will be some variation, right? When in the batch to batch, even in the product that you develop. So how much is, and where this variation comes from? All the physiochemical property that can change. Let's say you are manufacturing a protein based therapeutics. As you know, proteins are amenable for all sorts of stuff. Protein can aggregate, protein can degrade, protein can phase separate, protein can go through any chemical modification like oxidation or oxidation reduction, yes, that's right. What else? From glycosylation, amidation, all these can happen from the cell, but not all, kuch mein hi hoga. You know, it's a, so how much we can tolerate if there is amidation, oxidation, degradation, aggregation, all these. So that comes under risk assessment and defining the specification and limit. What is the limit that is acceptable? So there are some guidelines given by the manufacturing, but one can design it. We have to meet the specification, so I should be controlling from the beginning so that my process becomes robust and we get consistent product, right? So what are the requirements for developing standard methods for developing? First, you have to meet the requirement. What is the requirement for this product in terms of purity, specificity, toxicity, immunogenicity? That's a requirement that any product has to. The most important part is इतना डिटेल में रिसर्च कर लिए कि बहुत पैसा हमने डाल दिया, राइट? तो मार्केट में कहाँ वैल्यू पेश करोगे, क्योंकि फाइनली वन हैज टू, ए कमर्शियल ऑर्गेनाइजेशन हैज टू रिकवर द पैसा, राइट, व्हाट एवर दे पुट। इतना डिटेल में रिसर्च कर लिए हो डर डर के अब तो पता चला कि हमने इन्वेस्टमेंट बहुत ज्यादा कर दिया, अभी तो ऑल मनी विल गो इन देयर। दैट्स व्हाई आई एम सेइंग ईच प्रोसेस हैज टू बी वेल थॉट, वेल एग्जीक्यूटेड। Cost effectiveness is required because somebody is selling drug at a 50 rupees price. You cannot put it 500 rupees because you invested so much. Then you will not able to sell anything. So quality is very quality is important, extremely important, but you have to put it in such a fashion that it is cost effective, it is affordable. तो भारत में हम ड्रग बेच रहे हैं और ऐसा ड्रग बना दिए जो कि कितने लोग, माने बहुत कम लोग अफोर्ड करेंगे, तो उसका क्या फायदा है? कोई खरीदेगा नहीं। वैसे भी भारतीय जनता बहुत ही ज्यादा प्राइस सेंसिटिव है, ठीक है, राइट? हर स्कूटर खरीदते हैं तो पहला क्वेश्चन पूछते हैं कितना माइलेज देती है, राइट? तो वो माइलेज देने वाला कॉन्सेप्ट हैज टू बी, और कार खरीदोगे माइलेज देने वाला कॉन्सेप्ट हैज टू। सो एनी ड्रग कंपनी, फॉरगेट इवन ऑटो कंपनी एंड एनी कंपनी, हैज टू बी addressing the issue of cost, because finally you are going to deal with the market. So what, after putting all the resources, developing a fantastic drug, you have to place it so that it becomes a cost effective, and most important, it has to be robust. एक बात का ध्यान रख लो, कोई भी कंपनी कुछ भी प्रोडक्ट डेवलप करता है ना, उसको एक स्थिति में जिंदा रख सकता है, ये अगर तुम लोग कोई भी एंटरप्रेन्योर बनना चाहते हो लाइफ में, ये बात का ध्यान रखना, एंटरप्रेन्योर के ऊपर सबसे ज्यादा रिस्क रहता है और गेन भी सबसे ज्यादा हो सकता है। रिस्क क्या है? अपना तो पैसा लगा ही है, अपने साथ साथ कितनी जिंदगियों का वो मैनेज करता है, राइट। उसके सारे एम्प्लॉय के फैमिली उस पर निर्भर करते हैं। सो देयर इज ए लॉट्स ऑफ रिस्क विथ द एंटरप्रेन्योर हु इज स्टार्टिंग। एनी ऑफ you aspire to do that, you should be always careful. This is a very responsible job. Now robust means किसी भी शर्त में ऐसा नहीं होना चाहिए कि मैं बैंकक्रप्ट होकर बैठ जाऊं, सेबी का मेरे पास सो कॉल आ जाए, सुप्रीम कोर्ट में मैं टंगा जाऊं। You look at the side story of a very talented entrepreneur that we had for Jet Airways. नाम पता है उनका? नरेश गोयल, जेट एयरवेज, राइट। 5-7 साल पहले तक जेट एयरवेज वाज लीडिंग द एविएशन मार्केट इन इंडिया। आज क्या हालत है? सारे प्लेन बैठ गए, बेचारे वो जेल में हैं। अभी मैंने न्यूज पढ़ा, वो बोल रहे हैं कि अब आई जस्ट वांट टू, राइट। सो इट्स अ सैड स्टोरी। आई एम सेइंग इन एनी केस वन हैज टू प्लेस इट लाइक दैट, योर प्रोसेस, प्रोडक्ट, योर कंपनी रनिंग, एवरीथिंग हैज टू बी रोबस्ट, राइट। सो रोबस्टनेस इज इंपॉर्टेंट। स्केलेबिलिटी, अगर डिमांड बढ़ गया हाइड्रोक्सीक्लोरोक्विन का या डोलो 650 का, कैन इज बी स्केलेबल? So you have to think of scalable. Transferable, right? Scalable means like if COVID has come and say Bharat Biotech is making only 500 doses, can it be make 50,000 doses? So something like that. Can it be transferred if required, because you want to share your whatever with some money to other companies? Transferable. Then compliant and scientifically innovative. This is very, very important term. New product that you are making has to be scientifically innovative. When biosimilar you are making, it has to be little bit innovative. Then only you are positioned in the field. So for that, one has to really do the molecule assessment and candidate selections. So let's say we are generating a new antibody. So what are required? Antibody generation. Any idea? IgG molecule, right? Simple. How these different antibodies are made? And thought. मान लो तुम्हें एंटीबॉडी डिजाइन करना, अभी तो नरेंद्र ने बोला यू आर गोइंग टू मेक ए ड्रग, क्या सोच के डिजाइन शुरू करोगे? ममिता, सपोज यू आर डिजाइनिंग एन एंटीबॉडी, पहले किसके लिए डिजाइन करोगे? पहला क्वेश्चन ये है, व्हाट इज माय टारगेट? तो तुमने एक डिजीज पहचान लिया, उस डिजीज में किस रिसेप्टर से बाइंड करने वाला एंटीबॉडी हम डिजाइन करेंगे। ओके, देन व्हाट? तो तुम एंटीबॉडी या एंटीबॉडी ड्रग कंजुगेट बना रहे हो, व्हाट आर यू गोइंग टू मेक? आई सेड एंटीबॉडी, लेट्स डिजाइन एन एंटीबॉडी। तो डिजाइन एन एंटीबॉडी, मोनोक्लोनल एंटीबॉडी। Okay, clone design, and you want to also make say more effective, so you have to optimize little bit clone में कुछ variation करके we design a clone. Okay, so some 8-10 clones we have generated, hundreds of possible hits we generated, and you can do this nowadays using computational tool. AI is also coming to picture, docking and all. So lots of hits you generate, right, and show the effectiveness. Then you clone some of those, produce it and look at the bioactivity. इसमें से कौन बेहतर हो रहा है? So after this hundreds of hit, you come up with 6 to 10 मान लो monoclonal antibody we have taken. So how we came from hundreds to it? By looking at the bioactivity, how selective are they, and what is the stability and all those. So those parameters we are looking at that. Then we are doing sequence analysis of these. What are the sequence that has come, where it is binding, what is the binding efficiency. So all those process has led us optimizing from hundreds of the leads that we generated to 5 or 6 which we are going to take it forward. Now these are all done, and then we have to scale it up, right, we make it more. So, expression system you need, right. So, typically these are monoclonal antibody, cannot be done in bacteria. So, I have to take it in human cell, CHO cell. CHO cell we express, उसमें से कुछ होंगे कुछ नहीं होंगे, whatever, like CHO cell expression. CHO cell expression, because now we are expressing it in higher concentration, bulk and all those. So, CHO cell expression, some will be expressed, some will be not. Finally, whatever is better depending upon what is the cost in upstream, downstream, then quality, and finally the process to formulate stability, we have to check it. So, once we look at all the parameter after expressing it, which is expressing, how much it is expressing, how stable it is, when you concentrate it, because maps are not sold at a lower concentration, you have to concentrate and make it stable. So, after that you just end up with 1 or 2 or 3 whatever final candidate. So, these are the molecule assessment and candidate selections that we do, say, for monoclonal antibody design. Now, second is process development that happens in a development. So, now you find a molecules, दो तीन ये हैं. So, process development that happens in various distinct phase. So, we given this term like TPP, total product profile. What is total product profile? We are defining the parameters that are needed for a product and process to be developed. So, product we know and process what it takes. What are the process? उसको क्लोन के बाद, क्लोन सिलेक्शन के बाद अपस्ट्रीम, डाउनस्ट्रीम, purification, formulation, stability. So, all those we defined, and finally we came up with a TPP, total product profile. This is we define total product profile. उसके बाद we are going to do is assessing that molecule. So, molecule assessment. So, how molecular assessment is achieved? Because once we define the process, then we invoke QBD, quality by design, and right. So, quality by design, we have to make sure that once we design the product, there is not problem with a commercial issue, commercialization issue. What can be commercialization issue? Should be cost effective, right? Should be stable, so robust in terms of. So, these are the things that you have to look at when you do the molecule assessment of any molecule that we are developing. Then comes platform process. You know, platform process are कि हम एक क्या बोले मॉड्यूल डेवलप कर सकें which can be used for producing different say antibody. So, मान लो suppose you have made one antibody and that more or less is set in your company. Say you made a rituximab. Now, this process you know, right? This is my technology. For rituximab is say arthritis. You want to make a drug for breast cancer, for prostate cancer, and all those. Now, antibody, like you have developed the platform. So, this is the way I am going to do it. So, that if you change the molecule, it should be transferable with fine tuning, that one can fine tune and transfer it. That is a platform process that one can do it. So, leverage platform experience with a previous similar molecule or process to have a robust platform process developed. And finally is commercial process. So, optimize for yield, because this is very important. Always as a commercial organization one has to think कितना कम पैसे में कितना ज़्यादा हम डेवलप कर सकते हैं. ये बहुत ही इम्पोर्टेंट है, राइट. व्हेन बिकॉज यू आर स्केलिंग इट अप, इन्वेस्टमेंट शुड बी not that much, but product should be higher. So, optimize process for the high yield, robustness, stability, and then then it can be commercialization. So, some of the jargons that we are using, you can be like these are important to be known. Already I explained it. QBD is quality by design. How we are ensuring quality by designing the experiment that product is good quality. CQA, critical quality attributes. So, critical quality attribute of what are the important parameters for quality that drug must be having. like higher order structure should be same, aggregation state should be this much, binding should be this much, immunogenicity should be less than this much. So that's a critical quality attribute. Critical process parameter, right? In a process development, what are the parameters that one has to dissolve oxygen कितना होना चाहिए, pH कितना होना चाहिए, stacking flask में कितना stacking होना चाहिए. Multi attribute methods, right? When can you develop a method which monitors multiple attributes of a product? And then process analytical tool PAT, the cost of goods, GXP, good manufacturing protocol, good laboratory protocol, good documentation protocol. So this is called, or GCP, good clinical process. So GXP is all, right? GMP already we told, manufacturing में 5 process. What is GLP, good laboratory processes or good protocols? Good laboratory protocol. Documentation should be, that's a documentation protocol. Laboratory protocol क्या है? Instrument should be calibrated. कब खरीदा गया है, कब क्या स्टेटस है, केमिकल्स कब बनाए गए हैं, कितने लोग अपने बोतल पर लिखते हैं किस दिन हमने सॉल्यूशन बनाया और किस दिन एक्सपायर हो जाएगा, लिखा हुआ है क्या? दीज आर ऑल. लैब में कैसे आते हो, बाजार जाने जैसे या प्रॉपरली ड्रेस्ड अप होकर, राइट? व्हाट इज द प्रॉपर ड्रेस अप फॉर ए लैब? फुल पैंट्स, जूता, फुल स्लीव and then apron. So these are, this you learned in your practical also, but सभी लोग चप्पल ही पहन के इधर आते हैं, right? These are the good laboratory protocol. और ये सब अगर बचपन से ही सीखा जाए तो आगे समस्याएं कम होती हैं. Maintaining the protocol of lab, maintaining the documentation practice. I think Kamarnath emphasized a lot on documentation, right? Your lab notebook from day one should be written very nicely. Your protocol should be written very nicely. So that's a good documentation, good clinical process. We are not into clinical research, but if somebody is there, कितना डोज दिया गया, क्या क्या हुआ, सब everything has to be documented. That's a good clinical process. तो टारगेट प्रोडक्ट प्रोफाइल क्या क्या आने चाहिए उसमें. इफ यू आर से फॉर एन एग्जांपल वी आर डेवलपिंग ए मोनोक्लोनल एंटीबॉडी बेस्ड ड्रग, व्हाट ऑल इट टेक्स. सो से एफिनिटी ऑफ दिस मॉलिक्यूल, कितना एफिनिटी है, कितना बाइंडिंग होता है, कहां पर बाइंड करता है, वेयर इट बाइंड्स, बाइंडिंग प्रोफाइल, right? So binding epitope, binding site, that one has to look at the total product. What is the CDR range? CDR very good. तो उसका लेंथ कितना है, क्यों जरूरी है जानना? Very nice. So this one has to find it out, how many, what is the CDR length and what is the efficacy. PTM sites, where are the PTM sites and what are the possible PTMs that can happen? PTM is post translation modification, like phosphorylation, amidation, all these are PTMs. What kind of immunogenic response it can cause, right? Immunogenicity is extremely important. What kind of immunogenic response it can cause or it will not cause. ना करे तो अच्छा है, करेगा तो फिर कैसे रिस्क मैनेजमेंट करना है, that one has to look at. And finally we may thought of a nice product, but can it be manufactured? So manufacturability has to be done. So how you do that? So you have to find a cell line, right? You have to find a cell line जिसमें हम मैन्युफैक्चर कर सकते हैं. उसके अपस्ट्रीम के प्रोसेस क्या होंगे, डाउनस्ट्रीम के प्रोसेस क्या होंगे, फॉर्मुलेट कैसे होगा, what is the stability of this, and finally what kind of toxicology it can generate, right? So how we check it? So once product is there, we can check it by biophysical parameters, right? So biophysical, physiochemical potency, what kind of stability and potency it has. Affinity, for affinity say we design a molecule, Affinity one can say it, whether it is binding to receptor, what is the on rate, what is the off rate, right. A drug has to go and bind, do the work, but it should not stick, because this receptor will be doing multiple work, so it should be off rate also. So one has to find it out whether my drug is binding to the receptor with what affinity and going off with what affinity. Potency, the cross reactivity, and how we are going to administer the what is the route of administration. So many of the protein peptide based drug, we know that it's a given intravenous. So that's the thing. One can generate for testing to cut the cost, like one can be ready after this for phase 1. One can develop an ex vivo model for taking many of these, toxicity and all those can be done, or you can develop a organoid model. ये सब cost कम करने का, जैसे ही clinical trial में जाओगे बहुत पैसा लगता है, right? You are checking with human. So before we check with human, one can do all the test in a cellular model, right? Cellular model में उतना पैसा नहीं लगेगा. Whether drug is working, you can create a response readable outcome that whether drug is effective or not. Cellular model, ex vivo model, right? Organoid model, organoid you know, right? 3D culture we can make a organoid model we can check. Mouse model, all these are can be transgenic mouse model, one can do it, and then some designed mouse model one can do it. So these are the total product profile, all the way from safety, immunogenicity, one can go for phase 1, phase 2, phase 3. So this takes it manufacturability, toxicology, phase 1, phase 2, phase 3, and that's we are ready to do that. So who does it? It's a team. So many things cannot be done by one person. In a bigger company, it is done by like an organization, it's done by many people. Say let's start from here. There will be a team which will be developing a process. Process mein kya? There will be a team which is developing cell line, there is a team which is looking at upstream, downstream and formulation. Formulation wala banda सब चीज में एक्सपर्ट नहीं हो सकता, so he is just focused on formulating. His job is to make the protein stable at higher concentration. Similarly, cell line engineering, they're engineering the cell line for high yield production, developing a method for upstream, downstream and all those. And cell line engineering, if you are product developing, there is an analytical team which is analyzing the cell, the product and all those. So this is process development. Once process is set, now all the R&D is done for process on product, then you send it to manufacturing guys. Their job is to scale it up, make it higher, so that's a manufacturing. Finally, everything depends upon money. How much money is going, where you are generating money from, how are we accounting money for. So finance is very, very important. So, and finally once the product is done, there has to be a marketed, क्योंकि सारा पैसा उधर से ही आएगा फाइनेंस को देने के लिए. So marketing guy is there, and इन सबको कौन रेगुलेट करता है और कौन इनको कोऑर्डिनेट करता है? CEO of a company, right? उसके सर पे सबका बोझ है और सबका कोऑर्डिनेट करता है, right? So regulatory are there, program management is there, intellectual property is there, business development. These are not doing exact science, but they are very, very important. Because finally you want to develop a drug has to be approved by some regulatory agency. So there is a team which takes care of each of these steps so that it meets with the regulatory compliance. Now program management, right? So there has to be a guy, managers, trained managers, right? जो chaotic लोगों को, chaotic scientist लोगों को manage कर सके. वो लोग तो खोए रहते हैं सोचने में, लेकिन कोई आदमी तो चाहिए जो थोड़ा बहुत बता सके कि अब सर आज आपको बेंगलोर जाना है, इधर कुछ मीटिंग है आपकी, ये सब मीटिंग है. So program management is generally coordinating with many of these people. Intellectual, if you develop a new say intellectual property, it has to be filed and protected, क्योंकि सब लोग बना लिए तो फिर आपका फायदा क्या. सो एंड फाइनली कोई बंदा ऐसा भी होना चाहिए जो बिजनेस को डेवलप कर सके. अदरवाइज हम अपने एक छोटे से रूम में बैठ के फैंटास्टिक साइंस करते रहेंगे. वी कैन डू एक्सीलेंट साइंस, but फायदा, it's a commercial organization, so somebody should be there for BD, that's called BD, business development, right? अब इसमें थोड़ा सा जब मैन्युफैक्चरिंग जब आ जाता है, सो मैन्युफैक्चरिंग रिक्वायर्स इंजीनियरिंग एस्पेक्ट्स. द ड्रग सब्सटेंस इज API, drug product, formulated product, and if it can also be devices. जैसे मान लो इंसुलिन बनाए हैं इंजेक्ट करने के लिए, नीडल्स वगैरह चाहिए, तो यू हैव टू गिव दैट एज वेल. इंजीनियरिंग कैन कम विथ. देन MSAT. What is MSAT? It can be manufacturing, science and technology. So that's the MSAT, finance, marketing. So these are the typical organization of any biopharma company that we are dealing with. So this is the organization, this is how we are going to develop a drug. Now coming back to analytics of biologics. Analytics for biologics. दो इंटरेस्टिंग टर्म दिख रहे हैं. बायोलॉजिक्स हमने देख लिया, एनालिटिक्स भी नो. व्हाट आर द एनालिटिक्स? जो एनालिसिस कर सकते. व्हाई एनालिसिस इज रिक्वायर्ड? टू इंश्योर द प्रोडक्ट क्वालिटी, एफिकेसी, सेफ्टी एंड ऑल दैट. So we are basically looking at the naak, kaan, muh, aankh of any biological product that are going to taste it or visualize or hear or sense or smell or whatever for a biological product that is being manufactured, protein, ligand, whatever there. Right? So what comes in the analytics range? First is microscopy, just looking at what is happening. Microscopy comes like if you add a drug, whether it is going and binding to the receptor or not, can be easily done in a microscopy, light microscopy. Either a fluorescent tag dal diye, right, all what, and when it goes and binds, so DNA is colored with one color, this guy is colored with, both are co-localized, you know that it's binding. So one can do with a microscope. The another important things that is very well used in biopharma industry is mass spectroscopy. This is used for various aspects, checking the quality of the product. Quality means whether I have got the right product or not. You can do intact mass analysis. You can do the charge variant analysis, how much like a, you can do the digestion and find it out where is the PTMs has happened. You can do whether my mAb has come with some host cell protein or not. So these are the things that mass spectroscopy is used. Mass spectroscopy. One can do how the like a simple, one can, whatever we do in lab all are done. We can put the protein in and look at how the protein profile is changing, that's a proteomics. One can do how the gene profile is changing, that's a genomics, or transcripts are changing, transcriptomics. So genomics, transcriptomics, glycomics, immunomics, all sorts of omics are required for characterizing the biologics. So protein analysis, proteomics, gene analysis, genomics, transcript analysis, transcriptomics, glycomics. What is glycomics? Yes, glycan analysis of any of the product is, what is immunomics? What are the different level of immune response, right? What you measure? Cytokines, chemokines, IL, whatever, interleukin and all those can become in immunomics. No, it's not a mass spec. I am just saying that these are the analytics for this. Mass spec, उसके लिए ELISA kit वगैरह से करने पड़ेंगे, right? You can do the different analysis. एनिमल मॉडल में जा सकते हो, उससे पहले सेलुलर मॉडल में भी देख सकते हैं कि कितना इम्यून रिस्पॉन्स आया है. तो भाई एनिमल मॉडल्स तो यू कैन सर्टेनली यू हैव टू गो एंड शो इट प्री-क्लिनिकल, बट उससे पहले भी यू कैन अंडरस्टैंड इन द सेलुलर मॉडल साइटोकाइन कितने आए हैं, कीमोकाइन कैसे आए हैं, इंटरल्यूकिन कैसे हैं, ऑल दोज. फाइनली इफ यू इंजेक्ट इनटू एनिमल मॉडल, दिस हैज टू बी डन बाय क्वांटिटेटिंग व्हाट इज द इम्यून रिस्पॉन्स दैट इज फाउंड. हाँ, सेल एक ही टाइप के होंगे, बट वहां से शुरू कर सकते हैं फॉर 1 or 2 से इम्यून थिंग्स दैट इज हैपनिंग. हाँ बिल्कुल, समथिंग लाइक दैट. नाउ The bioanalytical methods are used for say binding study. We said no, K on and K off हमें निकालना है. तो कैसे कर सकते हैं K on and K off? Simple bind हो रहा है या नहीं? ELISA test, right? ELISA we can use it for saying binding happening or not. But by K on and K off can be done by various methods that typically use SPR, surface plasma resonance, BLI, whatever. All these techniques can be used for finding K on and K off. So here how SPR will work? Here you have a one substrate immobilized on a chip, pass the molecule, if binding happens, refractive index changes and you wash it, refractive index goes down. So this is K on, K off, right? So that's a ELISA or bilayer, BLI and many other things. Now cell based assay, like you can understand what kind of signal transduction is going on, what kind of transcriptional activation happening upon drug activation. Then one can go for animal model, transgenic model, a small and large animal, and finally if required epidemiology has been done. It's a physical, chemical and functional characterization of a novel biologics and biosimilar. What are the things required for novel biologics? Means first time, and biosimilars whatever copy, right? So first you have to make sure that primary structure remains same. How one can do it? Finding it out, intact mass without any degradation, if the mass remains same or not, intact mass. What is reduced mass? So yes, so say IgG, two chains are linked with disulfide bonds, उसको reduce कर दिए, oxidize कर दिए, reduce कर दिए, reduce करेंगे तो क्या होगा? चेन अलग हो जाएगा, उसके मास पर चेंज हो रहा है या नहीं. So that's a reduced mass analysis. So oxidize like intact mass without doing anything, mass spec से कर सकते हैं. Reduce mass again, reduce it and look at the mass spec whether the mass is changing or not. So that's giving us idea about primary structure. Another thing, इसको fragment करेंगे and that's a fingerprint analysis. One can do it. Then that's age old technique that was there, which is used for sequencing, peptide sequencer, right? Adamant degradation. एक एमिनो एसिड को डिग्रेड करेगा, उसका HPL से लेगा, दूसरे का प्रोफाइल मैच करके बता सकता है. पहला एलेनिन, दूसरा फिनाइलिन, तीसरा सेरीन, चौथा सिस्टीन, ऐसे. So that's also used for primary sequence analysis. So mass spec and N-terminal sequencer. Then disulfide mapping. Again, when you digest it, you can find it out. यहाँ disulfide बना है, यहाँ disulfide बना है. करेक्ट डाइसल्फाइड का बनना बहुत जरूरी है. करेक्ट डाइसल्फाइड का बनना बहुत जरूरी है. तो मास स्पेक दे सकता है, एनएमआर भी दे सकता है नोजी पैटर्न से, दैट इज यूज फॉर डाइसल्फाइड बॉन्ड फॉर्मेशन. एनएमआर में थोड़ा मेहनत लगता है बट निश्चित ही देगा. एंड मास स्पेक यू कैन डाइजेस्ट इट, फाइंड इट आउट व्हाट इज इट. सो दीज आर द थिंग्स फॉर डिटरमाइनिंग द प्राइमरी स्ट्रक्चर ऑफ ए प्रोटीन. नाउ as these biological drugs are prone, and these are basically biological drugs, that's why we don't manufacture them in bacterial cell, because they have a glycation pattern, and that's a glycans are important. So glycan patterns, glycoforms has to be done with a profiling. So one has to do again mass spec based on methods. You can do ESA, IMS, MALDI-TOF, HRMS can be used for this. Now monosaccharide analysis, fucoylations, sialic acid addition. Now people are also developing NMR based method to identify what kind of glycans is attached and where it is attached. So along with mass spec and NMR, there are lots of things that are developing. That's a glycan form. You have to make sure that glycans are attached in right amount at right position, which is matching. Say if you are making biosimilar, matching with your innovator product. So glycan analysis is again very important aspect. Then come higher order structural, because these are protein or peptide, has to be folded in a correct form. Then only they are effective. Folding of these protein peptide based drug is very important. Then only अगर सही फोल्ड होगा तभी तो जाकर बाइंड करेगा रिसेप्टर से, तभी तो रिस्पॉन्स देगा. अगर अनफोल्डेड हुआ तो बाइंड नहीं करेगा, इफेक्ट नहीं. So HOS is very important, that's called HOS, higher order structure. How you identify? Secondary से शुरू करेंगे. So how you identify? Protein secondary structure के लिए क्या करते हैं? far UVCD and FTIR, right? Similarly, tertiary structure के लिए near UVCD, fluorescence and all those. So these are low resolution technique. One can do it. Fourier transform NMR. Intrinsic and extrinsic. Intrinsic means if there are intrinsic fluor like tryptophan. Extrinsic, you add something which is fluorophore, either chemically attach it or just add it. You can get the fluorescence done for this. Then comes the protein stability. Right? If you are suppose making a biosimilar, agar innovator is stable at 60 degree, your product also has to be stable at 60 degree. How do you do? सब बायोफिजिक्स का एक्सपेरिमेंट इधर काम आता है. डिफरेंशियल स्कैनिंग कैलोरीमेट्री और टेम्परेचर बेस्ड सीडी और एनीथिंग दैट यू कैन डू टू शो द प्रोटीन स्टेबिलिटी. डिफरेंशियल स्कैनिंग कैलोरीमेट्री यू नो, एक इधर सेल होता है रेफरेंस सेल, एक सैंपल सेल होता है, हीट करते हैं और उसका हीट एब्जॉर्ब करते जाता है, देन इट शोज ए ट्रांजिशन. जहां मैक्सिमम होता है दैट इज टीएम. सो यू फाइंड इट आउट व्हाट इज द टीएम ऑफ दैट. One can use X-ray crystallography if the molecule has to be crystallized. You crystallize and get the structure. NMR spectroscopy, you don't, if it is not crystallizing, you can use in soluble form and use NMR spectroscopy for showing. If the protein is neither crystallizable nor soluble, you can use some of the low resolution technique like SACs. There are some molecules which forms a nanotube, like lanreotide is one of such. There are many molecules, it forms a nanotube. Abhi now you want to characterize those, right? So you can use it, SACs, small angle X-ray scattering, then particle imaging, the native mass spec, HDXMS. There are so many techniques that are used for getting the higher order structural analysis. Solid state NMR, yes. Perfect, you can do it, we are doing it. Solid state NMR is done. That's a developing technique and it is being done. So suppose protein is a microcrystalline, it cannot be solubilized. If you solubilize, you lose the structure, right? It is not making big crystal to uska X-ray kar sakte hain. Just do the solid state NMR. That's a perfect technique to be used. So these are various techniques for HOS. Then comes the purity, right? So how do you test purity? Again, all the experiment that we have done: size exclusion chromatography, charge variant analysis, reverse phase RP, reverse phase HPLC, right? Helix hydrophobic interaction, and all these glycation, oxidation, deamidation. So these basically is used for checking what kind of purity it has. So these assay has to be done. Now biological assay, so basically all sorts of assay you have to do. ELISA kit, the binding of your molecule to the receptor by various techniques. These are various techniques has to be done. So you analyze the data, interpret it. The idea is a comprehensive data set, years of work, comprehensive data set to establish the structural integrity, stability of a product. And this is required to convincingly prove it that we have a totality of evidence for a good drug manufacture. So basically totality of evidence is required from various experiments that we, series of experiments, series of biophysical experiments that is required for drug approval. So you see so many techniques, so many expertise is required and it's एक बंदे के बस में सारा चीज करना नहीं है. So that's how you need to collaborate, coordinate and comprehend the data. Where the world is moving, all these techniques are there. One technique in recent year becoming more popular because this requires less sample and get the high resolution structure. कभी कभी जरूरत पड़ सकती है. So I will just give you an idea. It's a cryo electron microscope. It is nowadays because of the optics, because of technology that has come up, sample requirement has reduced drastically and resolution has increased. So, firing of electron and detecting those response of those electrons is giving the high resolution picture up to angstrom level. That's called resolution revolution. कुछ साल पहले तक बल्बोग्राफी बोलते थे, सब कुछ एक बड़ा सा ऐसा बल्ब जैसा दिखता था, ब्लब जैसा दिखता था, बट अब एटॉमिक रेजोल्यूशन स्ट्रक्चर दिखने लगा. So you can see that's a cryo-EM that is developing and you can get this beautiful electron pattern, and that is required for, sometimes it is required for getting the higher order structure of it. So cryo-EM is one of the techniques that is what is looking forward going to solve. Like, you know how fast world is moving. कोविड वैक्सीन जो बना था उसका क्लिनिकल आइसोलेट लिए फटाफट उसका स्ट्रक्चर सॉल्व कर दिए 8-10 दिन में, और उसके बाद पता चला कि क्या बाइंड हो रहा है, कहाँ बाइंड हो रहा है, एपिटोप क्या है, उसके चलते mRNA वैक्सीन, डीएनए वैक्सीन, पेप्टाइड वैक्सीन जो भी बनना था बना. So this technique has really playing an important role in speeding up the drug discovery process, because unprecedented way structural resolution has improved with less sample and some effort. So cryo-EM is going to play a major role in coming days. Right. So there are some techniques which are not used often, like X-ray crystallography, electron microscopy, tomography, scan. They are not used very regularly. Sometimes once in a while you have to use it whenever required. But there are techniques which you need every day, right? So like a confocal microscopy, laser, because all the drug binding that is happening, stability CD fluorescence, TEM or TM वगैरह determine करने के, many of these techniques requires every day, right? Yeah, so flow cytometry and all those are required. Any question? हाँ, बट ड्रग किसी के लिए बना रहे हो, इंडियन पॉपुलेशन के लिए या यूएस पॉपुलेशन के लिए? क्वालिटी पर तो कॉम्प्रोमाइज नहीं कर सकते हो ना, तो इट हैज टू बी टाइटली मेंटेन. यूएस की ठीक है, कुछ स्पेशल रिक्वायरमेंट होगी, दैट इज जस्ट फ्यू परसेंटेज, राइट? मेजॉरिटी यू मेक इट, यू आर मेकिंग अ ड्रग एंड इट्स फॉर ह्यूमन. वैसा थोड़ा है कि इंडियन के लिए बना रहे हैं तो कम क्वालिटी का होगा. Essentially nobody thinks like that. Everyone makes a good quality drug. हाँ, थोड़े stringency हो सकता है, कम हो unregulated market के लिए, regulated market के लिए you go for very high stringency. But that's why that precisely this is the reason not too many companies are into novel drug, क्योंकि उसकी requirement बहुत ज़्यादा. Biosimilar में at least many things are known, right? So you are making a biosimilar for a known drug. Their requirement is not that much. See how many we have made it, the novel drugs? Not too many, few. The money requirement, if you look at the vastness of the research that needs to be done, is huge. तो देखो भाई, ये सवाल है कि आएगा क्यों. प्रॉब्लम तो है, लेकिन अगर प्रॉब्लम को सॉल्व नहीं करोगे तो कहां तक खरीदते फिरोगे. मैं एक जनरली दिखाता हूं एक पिक्चर कि 1950 में भारत एक बॉक्स दवा मंगाया था आजादी के बाद, ठीक है, और वो दवा को रिसीव करने के लिए अमृत कुमारी कौल या कोई भी अपना हेल्थ मिनिस्टर एयरपोर्ट पर गए थे. एक बॉक्स ड्रग पेंसिलिन का शायद था, मंगाया था. अगर हम नहीं मैन्युफैक्चर करेंगे तो अभी भी हर दवा को मुंह उठाकर देखना पड़ेगा वेस्ट की तरफ या कहीं और दूसरे देश की तरफ. राइट. तो हेल्थ वेट नहीं कर सकता है, भारत की 140 करोड़ की जनता है, अपनी दवा बनानी ही पड़ेगी, रिक्वायरमेंट है हेल्दी रखना। देखो भोजन, रोटी, कपड़ा और मकान, 3 बेसिक रिक्वायरमेंट है, हवा, पानी और भोजन, ये 3 चीज है। उसके बाद रोटी, कपड़ा, मकान वो हो गया। उसके बाद हेल्थ एंड एजुकेशन, दीज आर 2 इंपॉर्टेंट थिंग। यू हैव टू वांट टू कीप योर पीपल्स हेल्दी राइट ऑल द टाइम। कितने ड्रग के लिए तुम वेट करते रहोगे? 140 करोड़ जनता को हेल्दी रखने के लिए यू कैन नॉट वेट फॉर अदर्स टू थिंक। सो योर प्रॉब्लम कैन नॉट बी सॉल्व बाय अदर्स। अपना प्रॉब्लम हमें खुद ही सॉल्व करना पड़ेगा, राइट? तो अपना प्रॉब्लम सॉल्व करने के लिए किसी दधीची को तो हड्डियां गलाना पड़ेगा, तभी तो वज्र बनेगा। और कौन आएगा अपनी हड्डियां गलाने? और कोई नहीं आने वाला है, तुम्हें खुद ही गलाना पड़ेगा, तभी वज्र बनेगा। इसलिए जरूरत है कि प्रॉब्लम से भागो मत, फेस करो। और जो लोग फेस कर रहे हैं, ये नहीं है कि दे आर मेकिंग ओनली फॉर इंडिया, मेकिंग फॉर वर्ल्ड, बट इन द प्रोसेस ऑफ दैट मेकिंग, इंडियंस आर ऑल्सो गेटिंग गुड क्वालिटी ऑफ ड्रग एट ए अफोर्डेबल कॉस्ट। उसके रिवार्ड्स भी अच्छे हैं, बिकॉज यू आर मेकिंग। ठीक है, अमेरिका में नहीं बेचेंगे, यूरोप में नहीं बेचेंगे। अमेरिका और यूरोप की पॉपुलेशन ही कितनी है यार? 33 करोड़ अमेरिका है टोटल। बिहार यूपी मिला देंगे तो 33 करोड़ हो जाता है, राइट? तो हम अमेरिका के लिए नहीं बनाएंगे, बिहार यूपी के लिए तो बनाओ। यू सेल इट टू दोज पीपल, दे लेट देम बी हैप्पी एंड हेल्दी। तो यू हैव ए ह्यूज मार्केट हियर। एक भारत हो गया, बहुत सारे अफ्रीकन कंट्रीज हैं, बहुत सारे थर्ड वर्ल्ड कंट्री हैं, बहुत सारे कंट्री हैं जिनको सस्ती और अफोर्डेबल दवा चाहिए। उनके लिए वी हैव टू मेक इट। एंड बाय द प्रोसेस, केवल तुम जो टेंशन ले रहे हो, एफर्ट डाल रहे हो, ये केवल यही नहीं कि पैसा सूखते जा रहा, पैसा रिटर्न भी होगा। दैट्स द प्रोसेस। पीपल विल स्टार्ट पुटिंग मनी इन योर ड्रीम, बट आगे तो आना पड़ेगा। somebody has to take a lead। ये नहीं सोच सकते हैं कि सब अंबानी ही कर लेंगे, उनकी अपनी एक बैंडविड्थ है, लिमिटेशन है। So that's the requirement of this is the, I would say, need of the nation. We have to come forward and gear up for meeting the demand and expectation। ठीक है? सवाल यह है कि तुम देखो, हर टेक्निक का एक अपना लिमिटेशन होता है। बायोफिजिक्स में जैसे सिंपल एग्जांपल मैं समझाता हूं। एक हाथी का पिक्चर तुम्हें लेना है, ठीक है? हर टेक्निक, कोई टेक्निक अगर कल पूंछ को जूम करेगा तो क्या देखेगा? वो सांप भी इंटरप्रेट कर सकता है, रस्सी भी इंटरप्रेट कर सकता है और पूंछ भी कर सकता है। लेकिन तुम्हें बहुत सारी चीजों को कंबाइन करके देखनी पड़ती है। तो बायोफिजिकल टेक्निक इसलिए है कि सबका लिमिटेशन, जैसे डीएससी केवल टीएम देगा, एनएमआर एचओएस देगा, हायर ऑर्डर स्ट्रक्चर, लाइक एसपीआर करोगे तो बाइंडिंग एफिनिटी देगा। सो यू रिक्वायर मेनी ऑफ दीज टेक्निक्स टू बी डन। कितना इन्वॉल्व करना है, दैट कम्स इन व्हेन यू डिफाइन द टोटल प्रोडक्ट पैरामीटर्स एंड क्यू बाई डी, दैट इज रिक्वायर्ड कि हमें इतना जरूरी है, इतने सैंपल पर इतना पर दिखाना जरूरी है। सो all the time for all batch you don't have to do, but you have to collect the enough data that gives the totality of evidence that your product is of good quality। उस समय जमाना अलग था ना। वही टेक्निक जरूरी है, बट वैसा ही मॉलिक्यूल, बायोसिमिलर बना रहे हैं तो एग्जैक्टली वैसा ही मॉलिक्यूल होना चाहिए। उसने कैसे कैरेक्टराइज किया अब वो मैटर नहीं रहा, लेकिन मॉलिक्यूल वैसा ही होना चाहिए, ये मैटर है। क्योंकि उसने कम टेस्ट करके ज्यादा, उसने लार्जर सैंपल साइज पर क्लिनिकल टेस्ट किया है। जब बायोसिमिलर बना रहे हो तो तुम्हें उतना क्लिनिकल टेस्ट करने की जरूरत नहीं है, तो इसलिए प्रूव करो कि मॉलिक्यूल एग्जैक्टली वैसा ही बना है। हाँ, नहीं नहीं, रेजोल्यूशन ज्यादा हो गया, मॉलिक्यूल सेम होना चाहिए, बस इतना अभी बायोसिमिलर की डिमांड है कि तुम एग्जैक्टली सेम मॉलिक्यूल दिखाओ। इस अगर दिखा दिए तो शायद तुम्हें मिनिमम क्लिनिकल टेस्ट करने पड़ेंगे। उन्होंने बहुत ज्यादा क्लिनिकल टेस्ट करके मॉलिक्यूल यहां तक ले आए हैं।